Hello everyone and welcome to Ovarian Cancer Australia's Information and Support webinar. My name is Vanessa Alford. I'm part of the support team at OCA. I'd like to begin by acknowledging the traditional owners of the land that we're on and pay my respects to elders past, present and emerging. This webinar will focus on the latest developments in ovarian cancer treatment and research. We have three presenters who are going to share some valuable information with you. We're extremely grateful for the funding received for this webinar from AstraZeneca. The webinar is interactive, so over the next hour, you can ask questions to the presenters by posting a question in the Q&A chat box. Questions will be answered at the end of each speaker's presentation. Please note that presenters cannot address individual medical cases. If you have a particular question, please call the Ovarian Cancer Australia Information and Support line on 1300 660 334. I'm extremely privileged to introduce our presenters to you. Our first presenter is Professor Clare Scott, Joint Division Head of Clinical Translation and a Laboratory Head at the Walter and Eliza Hall Institute of Medical Research, and a Medical Oncologist at Peter MacCallum Cancer Centre, Royal Melbourne and Royal Women's Hospitals. Professor Scott will summarise what we know so far regarding HRD and why it matters to women. She will discuss what some of the options may be for Australia in the coming years. Our second speaker is Associate Professor Yoland Antill, a medical oncologist and cancer genetics specialist in Victoria with appointments at Cabrini, Monash Health and Frankston and Royal Melbourne Hospitals. Associate Professor Antill will provide an update on clinical treatments for ovarian cancer and give an overview on what we can look out for in the future. We will then hear from Rhonda Chapman, who was diagnosed with stage 3 high-grade serous ovarian cancer in September 2020. Rhonda has been on several clinical trials since her diagnosis and will share her experience of being involved in these trials. So I'd like to now hand over to the microphone, Professor Clare Scott. Thank you, Clare.
Thank you, Vanessa. Just doing the important handover. Hopefully you can all see my slides now in presenter mode. So it's a great privilege to be able to give this talk to you tonight. And as Vanessa said, I'm going to concentrate on HRD. What is it? Where we're going with it and why particularly it matters to women with ovarian cancer. It is a lot to cover in 15 minutes, so I'm going to whiz through some of the concepts, but I've provided a lot of detail on the slides. These are my speaker disclosures, and they do include research support from companies who make PARP and cell PARP inhibitors, such as AstraZeneca, and but I don't accept any honoraria for any advisory boards or presentations. So tonight I'm going to divide my talk up into 3 parts. So we're going to talk about biomarkers, what they are and how they assist us with clinical trials and with studying treatments. Then an overview of HRD itself and the tests and the challenges that those tests represent. And then some practical considerations based on where we are for HRD testing in clinical practice. So firstly, just to think about biomarkers. So they have horrible names: prognostic, predictive and exploratory. So we're just going to give some examples of each of those in ovarian cancer, so then we can see a bit of a difference. So if we think about BRCA1 and 2, and these are, initially discovered in the germline, but now we know that we can think of them in the tumour as well. So they provide prognostic information, and that means how a woman is going to do in terms of her cancer overall. We know that a woman with a BRCA1 or 2 mutation is probably going to have a cancer that will do slightly better than someone without a BRCA1 or 2 mutation, particularly for BRCA2. But we also know they're predictive of response, and that is to things like carboplatin chemotherapy and to the PARP inhibitor. So both BRCA germline and somatic are predictive. Now this HRD now has gotten to the point where we do believe it's a predictive biomarker, and I'll show you the guidelines for that. And then just as an example, is MSI or MMR relates to immunotherapy, and there are certain conditions where that is also a biomarker for response to immunotherapy for certain ovarian cancers. Then exploratory biomarkers include some of these other things listed here, but they're much less well understood, which is why they're exploratory and they haven't got to the stage of being predictive yet. So we're often these days using next generation sequencing here at diagnosis, and we would like to be doing this a lot more to stratify patients so that we know up front what sort of treatments they should have. But at the moment, this is not being done in the first line, apart from for BRCA1 and 2 in the tumour, and for a number of BRCA1 and 2 related genes in the germline. Why do we care about HRD at all, and what is it? So the HR part I'm going to describe first. So that stands for homologous recombination, which is just a really long way of describing a certain type of DNA repair. So all of our cells undergo damage all the time. And the reason why most people don't get more than one cancer in their lifetime, if they're going to get a cancer, is because our cells have evolved really sophisticated ways of repairing all types of DNA. So we have multiple different types of DNA repair pathways as well. But this HR pathway is really one of, or if not the most important, because it really has high fidelity. It does a very, very good job. It takes care of both strands. It's highly accurate. And when that is abnormal, so when certain genes from that pathway are missing, we call the pathway defective, and that's the D in HRD, so HR defective or an HR defect. And what we know about a PARP inhibitor is that it also plays into this type of repair. And if you get a double whammy or a double hit, you get a cell that already has a defect in homologous recombination, and you come in with a PARP inhibitor, which affects a number of different types of DNA repair in cells and in cancer cells. You end up getting cell death. Now, the good thing about this is if you've got a cell that is normal for HR repair, like other normal cells in the body, even in women who've inherited a BRCA1 or 2 mutation, the PARP inhibitor doesn't have the same effect, and cells survive and really don't give a hoot about the PARP inhibitor. So that's very good. It gives us what we call a therapeutic window, which means there's a much better chance that we can deliver drugs like PARP inhibitors to women without causing much in the way of side effects. So this whole concept that you need 2 hits is what is called synthetic lethality. So neither hit on its own is lethal, but both hits are. So we have some ESMO recommendations published in the Annals of Oncology, and I'll go through these in a moment, but this was the first time we really looked at the predictive capability of HRD for PARP inhibitors. So this again shows you what I was just describing in another kind of a pathway. So you've got the double strand break up the top, and all of these guys come in and repair. So these are proteins found in most cells in our body. And if everything's fine, HR proficient, you get this error-free DNA repair. But if there is a problem with one of the genes, particularly with BRCA1 or 2, or other genes, so either a mutation or silencing called methylation, you end up with a functional HR deficiency. Now, these next 2 slides are complicated, but I've just put them in case you want to see some of what we then talk about, particularly with the Myriad My Choice assay. So I'm highlighting that because at the moment that is the commercial test that most people choose to buy, to pay for if they're going to have testing. And it is in many ways, it's the gold standard elsewhere in the world as well. So it does 3 things, and I've defined them here, but you'll see them on a number of other slides, so don't feel like you have to remember it all here. There's a thing called LOH, which is how many gaps you get on one side of a gene, and it's across the whole genome, and we'll talk about that. There's this NTAI, but it's got a much better acronym called TAI, or just telomeric imbalance, telomeric allelic imbalance, and that is just showing that there are regions of chromosomes missing extending all the way to the telomere. And then large-scale transitions, and all of these add up together to give the GIS or genomic instability score. Now, separate to this, there was the HR Detect test worked out on whole genome sequencing, and this is the classic way we depict whole genome sequencing, which some of you may have heard of and some of you may have had performed on your cancer in the research setting. And this basically aligns all the chromosomes all the way around here in this Circos plot. And then you can see things like amplifications, deletions, and here we have translocations, so abnormal connections between chromosomes that shouldn't be connected. Now, when that is very noisy, and this one is very noisy, that really does look like an ovarian cancer that has HRD, because it's not been able to correct these before they've occurred, and it's resulted in all of these abnormalities. So this is just giving you a bit of an idea about what we're talking about when we talk about damage and the accumulation of damage. And signature 3 is the other way we describe this when we're talking about mutational signatures. So this has more precise information on it, but I'm not going to go through it. You're welcome to have my slides from Ovarian Cancer Australia, and then it's all there. So again, this algorithm just shows you the sorts of things that we think about when we're going to decide if a test is any good. We think it's a biomarker, there should be a phenotype, we should be able to predict a PAPI response, and then we should be able to stratify patients based on what we've learned here and use it in the clinic. So there were germline BRCA mutations originally, then we developed better data for tumour BRCA mutations, which included somatic BRCA mutations. Then we had some other genes, which I'll mention in a moment. Then we had these genomic scars. This GIS score is derived from the SCAR assays, and LOH is a slightly different example. So as a result of all of this, the updated guidelines, which were a year later in 2021, said that all patients with high-grade ovarian cancer should be tested for BRCA1 and 2, both germline and somatic, and patients with the BRCA mutations should receive 2 years of olaparib. But it also then said that testing for genomic instability, or HRD, is recommended because this identifies a subgroup of women who don't have a BRCA BRCA1 or 2 mutation, but who do derive benefit from the PARP inhibitor, and I'll show you some of that data in a sec. So patients with a positive HRD test and a partial or complete response to chemotherapy in the first line, with or without bevacizumab, should receive maintenance first-line treatment with a PARP inhibitor, and that could either be olaparib bevacizumab, according to the PALA-1 trial, or niraparib monotherapy. And patients who were HRD negative did not receive as much of a benefit, and the benefit was not considered to be clinically meaningful, and so therefore those patients were not advised to receive first-line therapy. And we could talk about that a little bit more later. So the HRD tests used in these trials were the Foundation Medicine, which was really mainly restricted to the LOH test, and tumour BRCA. The Myriad My Choice test that I've already mentioned, and My Choice Plus, which just includes a couple of other genes. So I just want to show you this pictorially. So these are the relapse trials, these are the first-line maintenance trials, and these are, just consider these as being major trials. These purple boxes are BRCA 1 and 2. And the further away this box is here from this line, the better. So you can see these pale pink. So these are the ones that weren't BRCA, but were HRD positive. They're not as far away as the dark purple. And this one is mixed with dark purple, so it doesn't count. You've got to go dark purple versus light purple, but it's still a long way away from the line. And the dotted line, which gives an idea of the error, is also a long way away. Now, having said that, here's the wild type. So why am I saying that the value isn't there? So here again, another trial, purple, light pink, but here you can see the green drifting in the first line towards the line. Purple, dark pink, and a bit of drift. Purple, dark pink, and a bit of drift. So I think what's clear is that for HRD, all of these light pink lines are a long way away from the line. So you might then ask, why has this not been approved in Australia in the first line? And one could say it's underway, but to what extent remains a little bit of a mystery. So this is another way of looking at all those clinical trials. And here we can see that the HRD, so BRCA wild type HRD, here again, we're looking at ratios or hazard ratios. And if it's less than 1, it's good. These are all way less than 1. So that is a positive thing. So we will leave that there and now just go and look at some of these implications. I don't have time to go through this in detail, but BRCA 1 and 2 is the key comparator. There are a couple of other genes. Some of you may have heard of these, RAD51C and D or PALB2, occasionally BRIP1. So certainly for these 3 extra genes, we believe that women with an ovarian cancer with those mutations should be on a PARP inhibitor, and we will do our level best to get access to a PARP inhibitor from a company such as AstraZeneca. And then there are a lot of other HRR genes that don't seem to have as much of an impact. The HRD tests do seem to predict for PARP response, and we would like to get access to the testing so that we can broaden the use of PARP inhibitors for women. So early treatment with a PARP inhibitor should be offered to every woman with high-grade serous ovarian cancer as early as possible, unless she develops platinum-resistant disease prior to having a PARP inhibitor. So sometimes in Australia, this HRD test is not done early, and so it is possible that a patient could develop PARP inhibitor platinum resistance at the end of their first line, so within 6 months, because they didn't receive first-line PARP inhibitor, and by the time they have platinum-resistant disease, it is less clear that a PARP inhibitor will help. So PARP inhibitor participation in trials and combination trials should certainly be encouraged, and I'll come back to that at the end. So this is just going to tell us a little bit about HRD tests and the challenge. I've talked about the My Choice test, which is the one we're going to talk about. This is a different test in the lab, which is really difficult and can't be done in the clinic. This tells you a bit about the whole genome sequencing scores that I've already talked about, LOH, which is the one that the Foundation Medicine test uses. And in fact, all of our tests do use LOH, but they use it in combination with these other two tests that I've already mentioned. So this now, is relevant for Australia. So this is the Illumina TSI 500 panel, which we're used to using in things like the MOST screening for most clinical trials. And they are now partnering with Myriad Genetics. So they're going to have the Myriad test involved in the TSI 500 panel. They've also developed a test for large rearrangements in BRCA1/2, which can sometimes be missed, and they've put all of that together. So this now is called this TSI 500 RUO, and it has the DNA variance DNA HRD test, as well as these BRCA large rearrangements. We don't really have that being easily sold in Australia yet, and I think there's still, I'm not clear what the price is, but that is something to watch. Now, the alternative to this, I'll just touch on very briefly, because it's not yet available easily in Australia, but this is called low-pass whole genome sequencing. So I gave you an introduction at the beginning into the concept of whole genome sequencing, because it can basically look with much more detail on a lot of these things. So this is called the Sophia Genetics HRD solution, and Stephen Fox at Peter Mac has been looking into this in a great deal of detail. And we also know that in this, they also do a capture panel that looks for some of the main genes as well, so you're not losing out on that. And this uses a proprietary deep learning algorithm to recognize genomic instability. So it is possible that this could be better than Myriad. There is some data to suggest that might be the case here, where the Sophia test was able to sort out a few false positives that Myriad were otherwise calling as HRD, which weren't. And this is the actual comparison of Myriad versus Sophia. But the broad take home is that there's a lot of agreement here. So if we were to say, which do we want in Australia? Do we want Sophia or do we want Myriad? Well, actually, we now probably do have a choice of either Sophia or the new Illumina combination test. There are some practical considerations, and that comes around what time should we be doing it? Should we be doing it straight early on? And I think we should. But I just want to show you this as my last piece. So this is called the Friends HRD Harmonization Project, and it's run out of the NCI in the US. And you can see a lot of names there of companies that are huge, including Myriad and other companies like Illumina. So what they did was they went and looked at all of these various tests and said, which ones actually work? That this was an in silico analysis. So that means they didn't do it for real, they did it in theory. When they did it in theory, it was a dog's breakfast. They couldn't tell what was good and what was bad. So then they decided to do it for real. So they had freshly extracted FFPE tumor samples from the TCGA. They extracted them at the NCI. They sent them to, I think, 14 different people like Myriad and Illumina and other providers. And the analysis is underway at the NCI, and the data is meant to be out by April. So that is really going to be important, because that's going to really tell us which of the HRD tests is likely to be the most useful. So just to recap, the Myriad My Choice is the reference standard, but the gold standard comes back to these original foci counting tests that aren't practical for the clinic. This HRD RUO test with the add-on to TSO 500 would be practical in Australia because Myriad is not licensed in Australia. We have to send tests overseas for that. And Sophia Genetics would be another alternative. So right now we're awaiting the outcome of the Friends Harmonisation project to tell us which tests might be best. We're awaiting PBAC and MSAC decisions, which I'm pretty sure are probably waiting to see what happens here as well. Once available, I would recommend HRD testing during first-line chemotherapy. We know that for BRCA mutation, we already have PARPI approved for 2 years in the first line until progression in second line, but we actually need to understand better whether patients should stop after 3, 5, 7 years or so. And in the BRCA wild-type HRD positive population, we know that many other countries already approve a Myriad test for treatment in the first line. And if, as a patient, you want to access that, you should talk with your oncologist about whether you can afford to pay for that or access it through research. There are a number of ways of doing that. And then it's 3 years of PARP inhibitor in the first line. But basically, we need trials of combined DNA damage response inhibitors, such as PARP inhibitors with other drugs that are out there, to actually make PARP inhibitors to work better, particularly in HRD BRCA wild-type cancers. So they're just some acknowledgements from people in my lab, and just to say that Stephen Fox has been a really big part of helping us understand HRD testing in Australia. And I think I'll leave it on that slide. Thank you.
Thank you very much, Claire, for providing all that valuable information. We just have one question. Someone has said many women are simply not aware of this testing, and this is incredibly frustrating. Why is this the case in Australia?
Basically because it costs $5,000, and until we have the test and a drug approved, it is a very difficult issue as to know whether to raise it. Even if one pays for the test and finds out that a PARP inhibitor would be recommended worldwide, it's also not something normally, if someone pays for a drug, you know, you can test after 2 to 3 cycles, after 2 to 3 months, you can do another CAT scan to see if it's working. And then at least you've only paid for 3 months. And then if you've got a response, sometimes you can actually try to get more drug out of the drug company eventually. But this is maintenance therapy. So to pay for 2 years worth, you know, or even 1 year's worth, it costs about $7,000 to $8,000 per month. So it's a lot of money to pay for maintenance therapy. It's hard to fundraise that amount of therapy. So even I, who am very proactive about fundraising, I find it hard to have that entire conversation. But, you know, it's time to be having this discussion. That's why we're having this webinar, and we would appreciate feedback. And there would also be opportunities for women to provide feedback to the PBAC and to MSAC.
And how soon do you think we will get testing in Australia?
I think we need to hear about the Friends Harmonisation project to see if there's a clear winner. If there's a clear winner with an HRD test, then our pathway will be easier. PBAC love clean data. They hate indecision and imprecision. So let's see what happens with that project. And I'm definitely close to the people who will know when the data's through. So we will find out straight away.
Thank you very much, Claire. We really appreciate you taking the time to join us on this webinar.
Thank you.
I'd like to now bring to the microphone our second speaker, Associate Professor Yoland Antill. Thanks, Yoland.
Hi, Vanessa. Thank you so much for inviting me to speak tonight. And I will try and share my slides successfully. Okay. So I am going to give you a little bit of an overview this evening on different clinical trials, thinking about different treatments for ovarian cancer other than PARP inhibitors. Claire's given an amazing overview, and I think that through the OCA platform, we've had a number of talks talking about PARP inhibitors, and I'm pretty sure most of those talks are still available. So we're really not going to focus on the use of PARP inhibitors in tonight's talk. We're going to take a step sideways. Okay, so just to remind everybody that there are different sorts of ovarian cancer. And the one thing to remember is that going forward, we are now separating out very different pathways of treatment according to the different types of ovarian cancer that there are. I'm just referring to epithelial ovarian cancer tonight. I'm not talking about germ cell, I'm not talking about the stromal cell or other rare types of ovarian cancer. So Claire's really focused her talk around the PARP inhibitors, which focus around the high-grade serous and probably high-grade endometrioid types, and to a much smaller extent, clear cell carcinomas. But this doesn't apply to low-grade serous carcinomas and definitely does not apply to the mucinous carcinoma group. We know that most women who are diagnosed with ovarian cancer are diagnosed at an advanced stage rather than localized. But we'll see as we move through the talk tonight that depending on the type of ovarian cancer that you have, that it may well, in fact, be an earlier diagnosis, but definitely share different drivers, the thing that caused the very first changes in cells to create the cancer in the first place. In particular, endometriosis has a role in risk for both clear cell as well as endometrioid. And the earliest startup lesions in high-grade mucinous or low-grade mucinous is not known, but we do know that there is a pathway between what we call borderline mucinous ovarian cancer through to low-grade and then into high-grade. But the very first and the risk factors for this rare subtype is not yet known. You can see down here that the genetic changes within the tumour and or genetic causes, hereditary-wise, are very different according to these subtypes. So in the past, we have lumped all of these tumours together and we've given them basically chemotherapy with platinum-based chemotherapy and hope for the best. But we know that the outcomes for these different subtypes varies. So the first part of the talk is going to think about immune therapy. And we've been really trying very hard to enable immune therapy to be utilised in ovarian cancer, but it's certainly lagging behind a number of other tumour types. There are 5 different sorts of cancer immunotherapy. So when you're talking about immune therapy, you might be talking about an immune checkpoint inhibitor, such as pembrolizumab, or you might be talking about monoclonal antibody therapy. And these are 2 types of immune therapy that we're going to spend a little bit of time about. But in ovarian cancer, there is just starting to be some very early trials looking at cellular therapy using the CAR-T approach to cancer therapies. And we will touch very briefly on the potential for cancer vaccine treatments. Oncolytic virus immune therapy is something that is being utilised in other cancers, but is a long way from being used in the ovarian cancer setting at this time. So if we think about Lynch syndrome or mismatch repair deficient ovarian cancer, it makes up around about perhaps 5% of all ovarian cancers. It's a pretty small proportion of all ovarian cancers. Mostly these cancers will be of endometrioid or clear cell histology rather than high-grade serous, and they are often diagnosed at a much earlier stage. We know from recent studies that around 40% of all of the what we call mismatch repair deficient ovarian cancers have what we call this high frequency of microsatellite instability. So we know that Lynch syndrome is one of the most common of hereditary cancer syndromes, but ovarian cancer makes up a small proportion of the cancer types associated with Lynch syndrome. More commonly, uterine cancers or bowel cancers are the ones that are seen. But the hallmark of these tumours is that the cells acquire multiple genetic changes, what we call a high mutational burden. And it's this lots and lots of genetic changes that can cause antigens to present to the immune system, and we often see immune cells infiltrating in and around the Lynch syndrome type cancers. But not all ovarian cancers will have this accumulated high genetic change, and so potentially not all cancers will therapeutically respond to immune therapies. We know that mismatch repair deficient tumours or microsatellite high tumours have access to immune therapies in other countries as single agent. If we look at some of the large studies that have incorporated lots of different types of tumours, ovarian tumours have been represented in small numbers and indeed show a relatively high response to single agent immune checkpoint inhibitor therapy. It's not yet available other than on a clinical study or for self-funding, but it's certainly a group of ovarian cancers that would potentially respond to immune checkpoint inhibitor therapy by itself without any other treatment. The clear cell ovarian subgroup are also a subgroup of ovarian cancers that seem to have a much bigger response to immune checkpoint inhibitor therapies when coupled with some other agents. If we look at some of the trials, early trials that have looked at immune checkpoint inhibitor therapy and immune checkpoint inhibitor therapy coupled with another immune stimulating therapy, the clear cell cancer subgroups were far more likely to respond than those other ovarian cancer subtypes. This may relate to the fact that these have an inflammatory background and have inflammatory cell infiltrate, but certainly there are a number of studies that are moving forward looking at immune therapies coupled with either a second immune therapy or alternatively with some small targeted agents for the treatment of this, again, smaller subset of ovarian cancers. Again, clear cell more commonly seen in the setting of endometriosis, Lynch syndrome, but interestingly is also the much more common subtype of ovarian cancer in amongst Asian ethnic subgroups. But we know for the majority of ovarian cancers, particularly high-grade serous and high-grade endometrioid, that single agent immune therapies really don't work very well. We know, however, that HRD tumours have a higher tumour mutational burden and therefore potentially have this signal to send to the immune system, and they express PD-1 and PD-L1, which is one of the or two of the biomarkers that have suggested immune sensitivity, but single agent has not been as successful as we've hoped. But if we combine immune checkpoint inhibitors with other agents, then the response to immune therapies seems to increase. Some of the other agents that can increase the likelihood of stimulating immune therapy response, so in other words, your own immune system to actually wake up and see the cancer cells there and attack the cancer cells, include chemotherapy, bevacizumab, which is an anti-blood vessel forming agent or an anti-angiogenic, PARP inhibitors, and other small molecule agents such as lenvatinib or other immune therapies such as CTLA antibodies. And this is just one example of a trial that looked at the combination of a PARP inhibitor with immune checkpoint inhibitor therapy. And you can see the different subgroups here. You can see that BRCA mutant tumours, HRD positive versus HRD negative, you've actually got a larger response across all of these subtypes when you combined the PARP inhibitor niraparib with the immune checkpoint inhibitor pembrolizumab. So this is just one example of where the research trials are going to try and get immune therapies to work in ovarian cancer. Another form of immune therapy are these monoclonal antibodies. And we're going to spend a little bit of time talking about what an antibody drug conjugate is. So mirvetuximab is an antibody drug conjugate. These, I might just move to my next slide. These are incredibly clever drugs where you have the antibody, which is attached to some chemotherapy. The antibody attaches to the receptor on the cell surface and is uptaken by the cell, or the chemotherapy is inserted into the cell, into the cancer cell that has the receptor for the antibody on. The chemotherapy itself causes cell destruction, and when the cell dies, it releases the chemotherapy to potentially affect the surrounding cells, what is known as a bystander effect. So mirvetuximab is an antibody drug conjugate focusing on the folate receptors when these are expressed on the surface of the tumour cells. You internalize the antibody drug conjugate, and then the active metabolites cause destruction of the cell. The destruction of the cell leaves this potential active drug to cause destruction of the surrounding cells. So the SORAYA study looked at mirvetuximab in patients who had folate receptor high expressing but carboplatin resistant ovarian cancer and found an overall response rate of around 30%, with a median duration response of around 8 months. And so these antibody drug conjugates are certainly making themselves known to the market. We do know that the overall response rate was seemingly higher if you had less lines of therapy prior to coming onto the clinical trial. But we also noticed, curiously, that if you'd had prior PARP inhibitor exposure, you had higher response rate. But certainly this is something coming into potentially to treatment options for advanced platinum-resistant ovarian cancer. But something to think about with all the antibody drug conjugates is that they often have very different side effects, treatment-related adverse events, than you would see with normal chemotherapies or PARP inhibitors or even immune therapies that we have seen in the past. In particular for mirvetuximab is the problem with eye toxicity, and this can be very, very dose limiting. It can stop people being able to continue on treatment. So each antibody drug conjugate has their own special set of unique toxicities. This is an up-and-coming other antibody drug conjugate, UpRi, and there's a whole series of trials that are now looking at the use of this agent with an overall response rate looking very favourable in the early studies, where you've got high expression of the NAPB2 receptors. So when you're using an antibody drug conjugate, you need to have the receptors on the surface of the cancer cell for the antibody drug conjugate to work, so that where the antibody is able to hook on to the tumour cell. So uplift and upgrade are currently in process, but the next plan is up next, and this will be a large phase 3 study in both platinum sensitive as well as recurrent but still platinum sensitive ovarian cancer as a maintenance therapy. So Claire's mentioned these early phase studies as first line setting of PARP inhibitors, and hopefully in the coming few years that we will see even more than the BRCA1 and BRCA2 mutated ovarian cancer patients being able to access PARP inhibitors, but the HR deficient tumours population as well. So what happens if we have a whole lot of women who've received PARP inhibitors in the first line setting? if the ovarian cancer does unfortunately become active again, could the tumours still be sensitive again in the second line? And one of the more recently published studies in 2021 was looking at this question. If you'd received previous PARP inhibitor, could the tumour respond to PARP inhibitor therapy again? And we did see a response to the PARP inhibitor. It wasn't as great as we'd seen in the first line setting. And we also know that the rate of hematological toxicities and some of the more serious hematological toxicities was more commonly seen when you received PARP inhibitors a second time. Remembering also that BRCA1 and BRCA2 carriers may be accessing PARP inhibitors in the maintenance setting in the breast cancer setting. So if they were to have received PARP inhibitor in the breast cancer setting and then require it for ovarian cancer, this is some indication that this may be tolerable and may be efficacious in that setting of ovarian cancer. And just to finish off, I thought I would talk about low-grade serous ovarian cancer treatment options. This is a rare type of ovarian cancer that affects often very young women. Surgery remains the foremost therapy, but these cancers, despite having high estrogen receptor expression, also often have frequent genetic changes in this what we call MAP kinase pathway. We know that these tumours are often more resistant to chemotherapy than high-grade serous tumours. We also know that they're often resistant to standard endocrine therapies after a short period of time, possibly relating to the activated mutations in this particular pathway. So this study looked at women with recurrent low-grade serous ovarian cancer who had previously received platinum therapy. They were biopsied and had retained their low-grade cellular status. So sometimes when these tumours become active again, they transfer into what we call a high-grade type. And these, in this study, they were looking at the use of an oral trametinib, which is a MEK inhibitor, compared to standard of care options, which included 3 different sorts of chemotherapy and 2 different sorts of oral hormonal therapy. MEK inhibitor is an oral treatment. So the overall response rate was around 26%. So this means that tumours either completely or partially responded, but a further approximately 60% remained stable and didn't progress for a minimum of at least 8 weeks. And this was favourable compared to the standard of care arm. They found a much higher response rate in those tumours that actually had a found genetic change in this MAP kinase pathway. And there were their unique side effects, as you would expect with the trametinib, that was somewhat similar to chemotherapy, but much more than endocrine therapy, but largely these were manageable. So trametinib is a treatment that will potentially overtake some of the lines of therapy down the track for recurrent low-grade serous ovarian cancer. We don't have access to trametinib in Australia yet, but I think the MEK inhibitors and MEK inhibitors coupled with other small molecule agents is certainly where trials are heading in this rare subgroup, but often affecting very young group of women. Thank you. And I'll stop sharing. Back to you, Vanessa.
Thank you very much, Yoland, for that extremely informative presentation. We have a couple of questions for you. The first one is, are there any upcoming trials for platinum-resistant cancers? Oh, you're on mute.
Yeah, sorry. Yes, that's a great question. And yes, there are a number of trials looking at these monoclonal antibody coupled, so these ADC trials. There's also immune coupled therapy trials, so not immune therapy by itself. And then there are also some really interesting studies looking at what we call cyclin E amplified or overexpressed cancers, and looking at small molecule agents that specifically target these tumours.
Great, thank you. And the last one is, what happens if I have a mixed type of ovarian cancer with serous and clear cell?
That's another really good question. We know that often tumours can have a mixture of histology types, but often there will be a predominant one. And when tumours relapse or become active again, it's often the only one sort rather than the combined sort that would recur. So treatment would need to be targeted towards that subtype. So that would be often why your doctor might require you to have a second biopsy, even though you had a surgery maybe 2 years ago and tissue was collected then.
Thank you. We'll just ask one last question. I know we are running short of time, but what are your thoughts on estrogen replacement therapy once chemo has been completed?
Oh, this is a really tricky question, because we know that often women are very young. They were premenopausal at the time of their surgery. And with cancers that are hormone receptor positive, this can often be a tricky question. I know that there have been some clinical trials, or not clinical trials, but some retrospective studies trying to answer this question. I think that for short periods of time, and particularly if symptoms are not tolerable, then the use of estrogen-only replacement in its lowest doses to enable control of symptoms is used at times, but not as standard of care.
Great. Thank you so much, Yoland. We really appreciate your time this evening. So I'd now like to hand over to our final speaker, Rhonda Chapman. Thank you, Rhonda. You're on mute.
Thanks, Vanessa, and thanks, everybody. And hi, everybody out there. This is hopefully a bit of an opportunity to get our head around some slightly different language, and I'll just share a quick couple of slides. There we go. So I'm somebody who has the triple whammy, platinum resistant, HRD negative, BRCA negative cancer, and diagnosed in September 2020, and I've had 2 lines of chemotherapy as well as been on a trial. I'll just introduce, so I'm just going to quickly run through basically what life is like on a clinical trial. So these are the 5 trials that I have been pre-screened for. You'll see this was the study, the Solace 2 one was the one of Clare's trials with the PARP inhibitor and immunotherapy. The Stafford Fox was a genetic screening trial, and this 3rd one that is in green, which is the one that I'm currently on, is one of the trials that Yoland was just talking about, which is an antibody drug conjugate looking at the anti-folate receptor alpha. There's one that I'm also being pre-screened for, which is another ADC trial, and the one that I didn't make, which was the cyclin E expression trial, that I was rejected for because I'd had too many lines of chemo, of treatment. I think the thing to think about these, as well as about the trials, is what it feels like, and in terms of why would you participate in a trial. This can probably look like I'm a bit of a trial junkie, but I guess the question is, why would you do it? And my question is why you wouldn't. It's good for research, but it's also been really good for me. The trials, you become eligible for a trial at a critical point in your treatment when maybe other options aren't working. And so it becomes an option to consider a possibility that you might be eligible. It is an interesting process, and I'll run through that process in terms of what it feels like to be considered for a trial. But I think the other thing, just for me personally, I'm a scientist by training, although I do have to say learning some of this new language has really tested my my old science muscle that I haven't done for a long time. But I think, you know, I firmly believe in the value of research and science for a broader reason, broader good. And so you know, I have been known to be a little bit harsh and say that if it, you know, if the trial doesn't work for me, I'm no worse off than I am now, but there's been a great benefit for research and for clinical understanding of ovarian cancer and how to treat it. But I think one of the biggest reasons, and not to, you know, suggest that I'm a martyr to this, is that it really offers me hope when I feel like I'm running out of treatment options, or hope for a different way to survive and to move through the, you know, being diagnosed with advanced cancer that, you know, is now platinum resistant. The other thing to notice is oh, sorry, I think you're seeing my notes as well, is that right?
We can see just on the right-hand side. Yeah, we can, Rhonda.
Oh, sorry about that. I don't know how it's having
okay.
gone through all of that before. So I think the other thing to notice is that I'm really privileged. You know, like I have, one of the reasons that I'm eligible for these trials is that I have not complicated other health issues. You know, I don't take medication. I don't have other health issues to manage. I don't have young kids at home. I'm self-employed. My lifestyle makes it a little bit easier to live and manage the challenges of being in a trial. So I think I'm just going to try and, we'll just keep moving through. So I think for me, what I wanted to talk about was also maybe the example of actually just being on a trial. So here's my current trial. It's an ADC trial working on the folate receptor alpha, the one that Yoland just described. And we only started this process on the 19th of January. So this is the trial by numbers. You can see it's moved pretty quickly. And since the 19th of January, I've had 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 different appointments as of yesterday. You can see what those appointments are. We start the whole pre-screening, calls, consultations. Do you meet the criteria to be eligible? Then do you satisfy all the medical tests so that you're not excluded, so that you can satisfy the criteria? So things like CT scans and eye tests, as Yoland described, being a critical factor for ADCs. You finally get a yes, and it starts pretty quickly. You'll notice that we got a yes on the 9th of February, and we started first treatment on the 13th of February. So things move really quickly. Because this is a first-in-human trial, there's a lot of screening and a lot of testing. So since the 13th, or since this process started, I've donated about 50 vials of blood to science. We've travelled about 1,800 kilometres backwards and forwards between regional Victoria and Melbourne, including some 3 separate train trips for a 20-minute appointment. I have absolutely no idea how much time I've spent doing this, doing the research, trying to get my head around the reading, asking lots of questions, etc., etc. And one of the benefits is that I've had 5 overnight stays with my mother-in-law, who, as of today, turns 99 years and 345 days. And so that's always a lovely benefit to do that. I'm just going to exit this and try and do it properly. I'm sorry about that. Let's share it better. Let's try this again. Is that better?
Yeah, that's perfect, Rhonda.
Sorry about that before. I shared the wrong screen, which we'd been through before. So I'll just quickly go back to that previous one. So that was just the slide in numbers. But I think that the thing to realise is, you know, there's got to be very clear, there's a commitment to contributing, to participating, and not to say I don't resent any of this at all. It's just good to have your head around the reality of what it's going to be like. And the time commitment. I remember one of the doctors at one of the pre-screening meetings asked me, so are you going to move to Melbourne because, you know, the amount of time we need to spend there. The other thing to note is that I know there's a big push to get more regional patients onto clinical trials. I am a regional patient, but, you know, I'm a regional patient who, again, is in a relative position of privilege because I'm regional in that I have relatively easy access to Melbourne. It's about an hour and a half, 2 hours into the hospital. For some people, you know, that's not a 5-hour trip. And so I think, you know, there's a whole lot of different factors to weigh up when you're making a kind of commitment to this time. The other thing I think is really important to talk about is the emotional side of being on a clinical trial. And people often talk about the roller coaster. The pre-screening, then all the exclusion tests. You know, we all know what scanxiety is like when you're leading up to having a scan, and that your world can turn on a penny, you know, based on the results of that scan. So, and people often talk about the anxiety, particularly recently where we had a lot of hope, me being considered and suggested for the Cyclone E trial, where I was rejected, and that was quite unexpected. And when you're feeling like that's one of the last options of treatment, it can be a pretty devastating roller coaster that you're on. And then suddenly there's a new trial and things move quickly. So I kind of described it not so much a roller coaster, but there's a whole process of the roller coaster that then shifts into a kind of a more stable routine, still ups and downs. And then the carousel. And I think the roller coaster is really that moment of when you're waiting to find out, are you on it? Have you asked all the right questions? My goodness, that can be hard. And I think Tim and I were reflecting the other night that as I've done more trials, I've got better at asking the questions, really. And do you get access to enough material, you know, of the prior research to actually know what questions to ask? Can you understand the research even when you read it? I've got a whole folder full of scientific papers that I would vow and declare I understand maybe 10 to 15% of them. So then you go through that whole decision-making process, and it's what I call the sliding doors moments. You know, they're just single little moments that can be a yes or a no, and that can make the world of difference. And then you're either back on the roller coaster again if you get a no and you have to go through all of that and then figure out what your options are, or you get a yes. You get moved into a treatment routine. You're carefully monitored. Oh my goodness, are you monitored? You're watched like a hawk, which for me, I really appreciate. I used to describe being on my earlier trial as being wrapped up in this gold safety net of care and people really asking lots of interesting questions. Took me a lot to learn of, you know, when they say, tell us any changes, let us know anything that happens. It's like, really, do you really want to know that? And so, you know, working with the trial team and the oncologist to be, what do you want to know? What do you want me to tell you? And that you've got usual up and downs in that process. So then the roller coaster becomes a bit more like a Ferris wheel, you know, steady and routine, but still ups and downs. Once you settle into the trial, you can move on to the carousel. You know, you can have an opportunity and you can settle into a bit of a routine. But because you're so closely monitored, that can flip really quickly back into being on the roller coaster, because you catch issues early and you need to respond, but you are back off the trial often and then back into treatment, which is what happened to me in my first trials. You know, it was a pretty quick decision and a bit unexpected, to be honest. So, you know, and that's the nature of the beast. It's not actually dissimilar to having ovarian cancer, but a little bit more intense, I would say. So then what do you do? Well, you know, I think one of the things I've learned is how to live a good life between treatments. And I don't know, Claire was my oncologist on my first trial, and I think we used to joke about how well I could manage as many holidays and as many great times away as I could in that 4-week or now 3-week window between treatment. It's really easy to be overwhelmed by the medical science of it, by the treatment, by the intensity of it, by all the, you know, the things that you need to do. So I think it takes a deliberate effort to not be consumed by what you're going through and the trial and all the demands of that. It takes conscious decision, and also the health and your fitness and ability, financial ability, a whole lot of different reasons, professional, work life, etc. But I also believe it becomes really important. Otherwise, everything in your life becomes medicalised. And for me personally, I'm not interested in that. So it's about, you know, finding that balance between, you know, look, sometimes it is hard work and sometimes you do need to lie low, but it's also about making sure you get out and do the things. On your screen is everything that I have managed to do in between 3 and 4 week treatment windows. It's possible. It needs support to do that. You need to be flexible. You need to, I think one of the big messages I have is that you've got to ask. you're like, the treatment team isn't going to say, so when would you like to go up and hike the Jatbula track in Katherine? If you want to do that and you're not quite sure how to do that and you think it's something that's good for you to be able to have a good life, you need to ask and be propositional about your ideas and about your needs. And my experience is that the medical and the trial team will work very, very hard to help make that happen for you. I know, because being a regional patient, you know, things like having scans and blood tests and scheduling all of those things, Peter Mac and certainly Cabrini try and do that the day of or the day before treatment, so you're not having to make 2 trips and sort of little those things. But I would suggest having that relationship and having those conversations and being your best advocate. My philosophy is that it never hurts to ask. The worst that they can say is no, and have a good reason to do that, and to be really organised and resourceful and willing to be flexible. You know, I mean, my attitude is book it. The worst that we have to do is cancel it. And at least if you book it, you know that you've got that possibility. What I haven't put up on that slide are the trips that we've had to cancel, because that's just too boring to contemplate. And of course, none of this happens without a village of support. I have been, and you know, it makes me a bit teary to think about how incredibly dedicated and caring the medical people are who look after me. You can hear it in my voice. Just absolutely everybody, and just their best interests, who really work hard for you. But also family and friends, both near and far, who hold me in their hearts and stay connected and, you know, do the best that they can to support me through this. And then, of course, there's the people who are there every day. Not only Tim and the rat bad dog Carlotta, but also my crew of Ronnie Rescue Rangers. And the Ronnie Rescue Rangers are a conversation for another day. Thank you. That's me done.
Well, thank you so much, Rhonda, for sharing, you know, your journey, your experience of being on clinical trials. I know everyone on this webinar would be so appreciative, just, you know, for being so open and honest and willing to share your story. So a big, big thank you. We have a couple of questions. The first one is, have you regretted signing up to a clinical trial?
No, not once. I'm never sure I've asked all the right questions, but no, not a single one. And I think, you know, and I feel incredibly privileged to be doing one of the hot off the press trials that Yoland was talking about. And so, you know, it's scary, absolutely scary, you know, the risks are there. But I think the important thing to know is that there's a get out of jail clause really easily. You know, you're there as a voluntary participant, and you can leave as soon as it doesn't suit you. And you know, you're not signing anything away. And so you just need to, my whole attitude is to work with the clinical team, the oncologist and the trial coordinators and the nurses as colleagues in a collaborative way, and that you just keep those lines of communication open.
That's wonderful. Thank you. And our last question, I know we've gone a little bit over time, but is there anything that would have made the decision to sign up for a clinical trial any easier?
Oh, yeah. And I'm, you know, I may have mentioned this to Claire, but I so have a little bee in my bonnet about the summary page that goes on the top of those bloody 27-page consent forms that, man, oh man. So I think, I know it's hard language to get around. As you've seen, it's complex science. It's really cutting edge. It really, you know, that for me, I get that they're written by the clinical researchers who know what they're trying to test and study and have trial participants for. But I think I'd really like to have a couple of pages on the front of it that are written from the perspective of the trial participant and saying what questions would you be helpful to ask? What's the so what in this for you? Claire's nodding because I've rabbited on about this in the past, but yeah, so I think, you know, I think it's that whole communication about it, because, you know, I have, you know, long dormant, but a science muscle somewhere there in my brain, but I found it really, really hard to kind of navigate and figure out the questions and what I need to know.
We have 2 more questions and then we'll have to leave it there, just conscious of time. One is, have you experienced any neuropathy? And the other one is, do you know how many clinical trials a patient can go through or is allowed to go through?
Well, look, I'm up to screening for number 5, so I have no idea about that. But, you know, I'll hand that over to Clare and Yoland. I have had neuropathy and from my first round of chemo, and thanks to my support crew, keeping me walking, keeping me moving, keeping me exercise. And I'm just going to put a plug in, Clare, a little bit of cold swimming. I reckon I've managed to make, it's pretty much gone away. I don't think I had a particularly severe case, though.
She really does mean cold water swimming. Very impressive.
Clare, did you want to answer that question just while you're
there?
there?
So I mean, there is no limit as to the number of trials you can go on, but trials have different sorts of criteria. So there will be some trials where you can only have had a certain number of prior lines of therapy, but while ever you are well, and you know, if your doctor doesn't have the right sort of trials, we actually all ask our colleagues. So if I don't have the right trials, I will ask colleagues at another hospital if they have, and that's what, you know, has happened to Rhonda. She has gone from Peter Mac to Cabrini, and by the way, Cabrini take patients who are public patients. They don't make you pay, even though it's a private hospital, so it's part of our network. So it really doesn't matter where you are. You can always have that conversation with your doctor and ask to have your case referred in to have it be discussed for other trials. We have, certainly if you're unwell, there may be the time where you are too unwell to go on a clinical trial, and your doctor will know you best. And if you are given that advice, then I guess you could ask for a 1-second opinion. But if 2 oncologists agree, then that's likely the case. And that's about protection. You know, it's about, you've heard trials are a lot of hard work and there are risks and toxicities involved. And those are really the protective features that make your oncologist say, I just really don't think a trial is for you. But up until that point, you can keep looking for a trial that suits you at that point in time. And there's never only one trial. There's just the trial that ends up being right for you at that point in time, and otherwise keep looking.
Fantastic. Thank you. Wow, so much important and valuable information has been shared over the last hour. I want to say a big, big thank you to our presenters. Just got a couple of slides that I'll share as I do. So big, big thank you again to Professor Clare Scott, Associate Professor Yoland Antill and Rhonda Chapman for giving up your time this evening and sharing so much important and valuable information with us. Thank you everyone for being on this webinar. We really hope that you got a lot out of it. The recording of this webinar will be posted on our website in the next couple of weeks. You will also receive an email in the next couple of days with a recording to the webinar and also with an evaluation survey. So if you could just take a moment, it will take a couple of minutes just to complete that survey, because your feedback is very important to us and it does help us to plan future webinars. If you have any other questions that you would like addressed or you feel that you require extra support, please don't hesitate to contact our information and support line on 1300 660 334. Thanks again, everyone. We hope to see you for our next webinar in a few months' time. Enjoy your evening. Goodbye.